Today we are going to discuss the fundamentals of human skeletal muscle. We'll begin today's lesson by discussing the basic properties of human skeletal muscle. I'll then move on to identifying the line of pull and angle of attachment of muscles and how that affects the movement of joints. Then we'll discuss the arrangement of muscle fibers and their relationship to connective tissue structures that surround them. Then we will zoom in and discuss the specific anatomy of an individual sarcomere, which is the contractile element of human skeletal muscle. And finally, we will discuss the sliding filament theory. So beginning with the properties of skeletal muscle tissue, the first item on my list is excitability. Excitability is the ability of a cell to respond to a stimulus by altering its membrane potential. And the way that it alters its membrane potential is by allowing various charged ions to cross the membrane. Related to excitability is the idea of conductivity. Conductivity allows that change in membrane potential to spread over the cell membrane, conducting that signal all over the cell. Contractility is the ability of a muscle cell to develop tension. And when it develops tension, this can either lead to a shortening contraction of a muscle contracting and remaining the same length, or even a muscle contracting and being lengthened. So the idea again of contractility is just the ability to develop tension within the skeletal muscle cell. Extensibility is the ability of a muscle cell to be passively stretched. So when the muscle cell is not being actively contracted and is not developing tension, you can move a joint through a range of motion that passively stretches the muscle. That is extensibility. Jumping off of extensibility is the idea of elasticity. Elasticity is when a muscle cell is stretched past its resting length. There are various connective tissue structures that surround the cell, as well as some proteins within the cell itself that will want to cause the skeletal muscle cell to rebound back to its resting length. Now that we understand the basic properties of skeletal muscle tissue, let's translate it back into human movement. So the first major idea that I want you to have from this segment is a muscle's line of pull. The line of pull of a muscle is a straight line from the muscle's origin to the muscle's insertion. And when a muscle develops tension, it will pull along this straight line. The resulting movement that takes place is due not only to the line of pull of the muscle, but also the type of joint that is being crossed and any support structures such as ligaments and labrum that might prevent movement at that joint. Now, in some muscles, the line of pull can be very clear, such as the rectus femoris line of pull that was displayed in the previous slide. However, lines of pull can be dynamic based on the muscle and joint that we are talking about. So there's two general ways that a line of pull can be dynamic. The first way is based on joint position. So I have a little 1 next to joint position because I want to link that to the image that is labeled number 1 below, which shows the pectoralis major muscle. Now, if you remember back to your anatomy class, you learned that the pectoralis major muscle flexes the glenohumeral joint, the shoulder, and also horizontally adducts the glenohumeral joint, such as doing a push-up. What is something that no one ever learns in anatomy class is that the pectoralis major can also extend the shoulder joint if the shoulder is beginning in a flexed position. So I want you all to take a moment right now as you're listening to this video and put one hand up on the desk or table in front of you so that your shoulder is in a flexed position and your hand is resting on the desk. Now I want you to take your other hand and dig into your deltopectoral groove where you can feel your pectoralis major muscle. And I want you to begin with the pectoralis major relaxed. Now I want you to try and extend your shoulder. So push down on the table. And what most of you should feel, or all of you should feel, is the pectoralis major becomes tense. that should tell you that the pectoralis major is attempting to extend the shoulder joint. So therefore, the pectoralis major can either flex the shoulder or extend the shoulder based on the starting position of the joint. So there's one way that the line of pull can be dynamic. The other way that the line of pull can be dynamic is anytime that we're looking at anatomical pulleys. So for this, I have it linked to images 2 and 3 on the right of this slide. So image 2 is showing the psoas major muscle. And when we stand in this normal anatomical position shown here, where the hip joint is in a neutral position, if the psoas major were to contract, it would want to pull the femur directly forward. But as the femur begins to flex at the hip, the line of pull of the psoas major muscle rolls around and follows the femur as we move the hip into a flexed position. So throughout the range of motion, so joint position you can think of again, but as we move throughout the range of motion of an anatomical pulley, you see the line of pull change. It's not a straight line from origin to insertion anymore. And in the image down below, we see the knee, where in a flexed position, we form a pulley with the quadriceps muscles as they cross over the patella to insert onto the tibial tuberosity. So on the left side, where the knee is in a greater flexed position, you can see that the angle between the femur and the line of pull of the muscle is greater than when the knee is in a more extended position, such as on the right. You can see that angle between the femoral shaft coming down and the line of pull of the quadriceps muscles becomes less. So again, that's an example of an anatomical pulley and the changing of the line of pull throughout the range of motion. The next concept that I would like to introduce is the angle of attachment. Angle of attachment refers to the angle between the line of pull of the muscle and the bone that it's inserting onto. And the angle of attachment has been a challenging concept for students in the past, so I would actually like to begin by giving sort of a real-life example, that is pulling a door open, and then I will link it to how it works out in kinesiology specifically. So if we think about opening a door, and you see my display door down below in blue with the hinges in green and the doorknob in orange, if I were to grab onto that doorknob and pull on it to open the door, what would be the best direction for me to pull on that doorknob in order to maximize the rotation or the swinging open of the door? So let's look at 3 different examples. So option 1 is if I were to grab onto the doorknob and pull in the direction of this arrow. Now if I did that, the angle between the door and the direction of the force that I'm pulling is very, very small. And if we think about this force as an X and Y component, or how much of the force would be pulling directly in line with the door and how much of the force would be pulling perpendicular to the door, I've added in those 2 directions of force here in purple and in red. Now, the component that's in purple that's directly perpendicular to the door, that's the component of the force that would actually cause rotation of the door, causing the door to swing open. The red component, that's the component of the force that would essentially be pulling the door into the hinges. So this would not be an optimal direction for me to pull on the door, because most of the force that I'm pulling with would be pulling the door into the hinges and not actually opening the door, causing the door to swing open. So that's option 1. If we look at option 2, what if I were to grab onto the doorknob and pull it in this direction? Well, now in this case, we have a very large angle between the door and the direction of force that I'm pulling. And if we break it down again into the X and Y components, you can see that the component that would cause the door to rotate open is in purple and is again very small. The component that is shown in red would be how much I'm pulling on the door, like pulling it away from the hinges. And that's essentially, again, wasted force. It's not going into actually opening the door. So this would not be an optimal direction to open the door. And if we look at one last option, a third option, what if I were to pull on the doorknob directly perpendicular to the door? Well, in this case, we have a perfect 90-degree angle of the direction of force that I'm pulling in relation to the door. And what that actually means is that all of my force is going into rotating the door open, the purple vector. You see there's no red force vector in this image because I'm not wasting any of my force. So if you think about the thousands of times that you have opened a door in your life, when you turn the doorknob and pull, you tend to pull directly perpendicular to the door. And I want you to actually spend a moment and experiment with this on your own just to wrap your head around it, pulling on a doorknob from multiple different directions and seeing the result as you open the door. So now let's translate this idea of opening a door to the angle of attachment of a muscle as it acts on a bone to cause rotation. So the first thing is I want to just orient you to the images that we're looking at here. So what we see is the upper limb. So we see the scapula and clavicle, the humerus, the radius and ulna, and the bones of the hand. And we are going to look at the action taking place at the elbow joint. So if we begin with the image on the left, the first thing I want to do is just draw this black line to show you the lever, the segment that's moving. This lever you can liken to the door in my previous example. Okay, this is the thing that's actually going to be rotating. The elbow would be acting like the hinge. Now the muscle, the biceps brachii in this example, can contract and develop tension and pull in its direct line of pull. And the difference between the muscle's line of pull and the lever is the angle of attachment. So in this image on the left, we see we have a fairly small angle of attachment. Now, just like we did with the door, if we look at that force that's being generated by the muscle and we break it into sort of an X and Y component, the component that is directly perpendicular to the lever, that's the component that would actually cause the anabrachium and hand to rotate at the elbow, like opening the door. That's what's called the rotary force. This is the force that actually produces movement of the joint. At the same time, we have the force that would be, in the example of the door, like pulling the door into its hinges. So in this case, this would be considered a stabilizing force because we are pulling the radius and ulna into the humerus. So it would be compressing that joint and stabilizing the joint. So the takeaway again from this image is that of the force that the muscle is producing, based on its angle of attachment, some of it is going into stabilizing the joint, some of it is going into actually producing our desired movement. Now, if we look at the image on the right, again, here is the lever drawn in. If I draw in the force vector for the muscle, we now have a very large angle of attachment. And if we break that force of the muscle into the X and Y component, we see the rotary force being directly perpendicular to the lever that is the anabrachium and hand. So that would be the component that would actually produce movement of the elbow. And now we have the other component of our force vector, which is directly parallel to our lever. And in this case, it's what we would call a dislocating force. So this was like the second example of opening the door, where we were pulling the door away from its hinges, like you're trying to pull the door off its hinges. So in this case, we're pulling the radius and ulna away from the humerus, hence the name a dislocating force. Now, just like the example of the door, and I don't have a clear picture on this image, but the optimal angle of attachment where the force that the muscle is producing is going directly into moving the joint would be 90 degrees, just like the 90-degree angle of force when we pull open a door. So that image is not shown here, but that would be a good takeaway. Okay, so based on the angle of attachment, we have more or less of the force that the muscle is producing going into actually moving the joint. And the most optimal angle for moving the joint is 90 degrees in relation to the bone that the muscle is inserting onto. Next, we're going to look at a variety of different muscle fiber orientations or arrangements within skeletal muscle. So the fiber arrangement is based on how the muscle fibers run in relation to the tendon. And we have two general categories. The first category is where the muscle fibers line up with the tendon. And the second category is where the muscle fibers attach to the tendon at more of an angle. So let's focus first on the groupings where the muscle fibers line up with the tendon. So the first classification are longitudinal muscles. These are muscles such as the sartorius of the lower limb. So the sartorius is a very long muscle, one of the longest muscles in the human body, and you have individual muscle fibers that are 2 to 3 feet in length depending on how tall an individual is. And all of those muscle fibers are lined up parallel with each other. The next classification are quadrilateral. Unfortunately, we do not see an example of this in the image on the right, but quadrilateral essentially means four-sided. So you can think of the quadratus lumborum or the quadratus plantae. In this arrangement, again, the muscle fibers are running from origin to insertion in a direct line parallel to each other. Next, we have triangular or convergent muscles such as the pectoralis major or the latissimus dorsi. For these muscles, they have a very broad origin and a very narrow insertion. So they're going from that broad origin and those muscle fibers are converging on the insertion. That being said, the general orientation of the muscle fibers to the tendon is still lined up or parallel to the tendon. And the final grouping within this overall category is fusiform muscles. Fusiform means that the middle or the belly of the muscle is larger in cross-sectional diameter than the two ends of the muscle. So an example of this would be the biceps brachii, where the proximal and distal ends are quite narrow, but the very middle belly section of the muscle is very large. Again, notice how the muscle fibers line up with the tendon from origin to insertion. The second overall category of muscle fiber arrangement are known as the pennate muscles. Pennate comes from the Latin term pennum, which means feather. This is because these muscles have an angle of attachment onto the muscle tendon itself. If you look at the bottom left image here, I've drawn in this white portion that would be the tendon of a muscle, and then the muscle cells you can see are coming in at an angle to attach to that tendon, giving it the appearance of, in this case, half of a feather. So let's look at each of the three types of pennate muscles. The first are the unipennate muscles. This is specifically what the diagram in the bottom left image is showing. If you look over on the picture to the right, it's showing the extensor digitorum longus of the crus. So unipennate muscles, like one side of a feather, are found predominantly in the antebrachium and the crus, and many of the muscles of the antebrachium and crus are unipennate muscles. Next, we have bipennate. Bipennate means that we have pennation on both sides of the tendon, so this would have the appearance of a complete feather. Bipennate muscles are again found throughout the antebrachium and crus, and the example to the right shows the rectus femoris, one of the quadriceps muscles on the anterior thigh, as a bipennate muscle. And the third and final type of pennate muscle is called multipennate. A multipennate muscle is like multiple other pennate muscles combined where they converge onto a single tendon. So the best example is the deltoid muscle, where it has an anterior, a medial, and a posterior head, where those heads are pennate in and of themselves, but they all converge together into a common tendon that inserts onto the deltoid tuberosity. Another example of a multipennate muscle is the subscapularis. And all of you should take a moment to Google the subscapularis muscle. And if you see it on a cadaver example, you can see the multiple sections of pennate muscle that are all coming together to form a single insertion point. Now that we've looked at the macroscopic arrangement of various muscles, let's begin zooming in. And the first thing we're going to do is look at the arrangement of the connective tissue structures to the actual muscle cells themselves. So for those of you that took anatomy here at the University of Utah, this image may look familiar from Mark Nielsen's anatomy class. If we look first at the muscle cells, which are shown here in red columnar structures. So like this right here would be an individual muscle cell. Here we see muscle cell, muscle cell, muscle cell. Notice how all of those muscle cells are wrapped in what's color-coded here as yellow. This yellow is endomysium. Endomysium is a loose collagenous connective tissue that wraps around the individual muscle fibers. Now, around those individual muscle fibers and their endomysium, we package groups of muscle fibers with another connective tissue wrapping that is coded here in light blue. This is called paramysium. Paramysium is going to package individual muscle cells and their endomysium into contractile bundles known as fascicles. So that would be the paramysium. Paramysium is a dense, irregular collagenous connective tissue. We then have groups of fascicles. So here's our first fascicle, but each one of these is one fascicle, a second fascicle, a third, a fourth. And those bundles, those fascicles, are then wrapped by another layer of connective tissue shown here in green, which is called epimysium. The epimysium is going to package fascicles into individual muscles. Again, epimysium is made of dense, irregular collagenous connective tissue. Finally, shown here in purple, wrapping around entire limbs and groups of muscles, we have what's called fascia. Fascia is a dense, irregular collagenous connective tissue. So what I want you to notice is all of these layers, other than the actual muscle cell itself, are made of a connective tissue with the component of collagen. The endomysium is a loose collagenous connective tissue, whereas the paramysium, epimysium, and fascia are a dense collagenous connective tissue. And now the next thing we're going to do is we're going to translate these connective tissues that we see within the muscle belly itself to the tendon and how the tendon is going to attach to the bone. In this image, we're now seeing a muscle cut in a longitudinal section, but I want you to notice that it is color-coded the same as the muscle that we saw in cross-section in the image before. So the red portions are the muscle cells themselves, the myocytes. You'll notice each of those is wrapped in yellow, which would be their endomysium. Groupings of myocytes in their endomysium are then packaged into fascicles by a wrapping of paramysium in blue. And then the entire muscle is wrapped with green, which is the epimysium. Now, the purpose of this image is to show you how the muscle cells and the connective tissue structures look in the muscle belly, in the muscle tendon, and then how the tendon actually attaches to our bones. So, in simple terms, a muscle tendon is just the continuation of all of those connective tissue layers once the myocytes have ended. So if this is the muscle belly here, you see that the muscle cells themselves end right about here. So everything from where the muscle cells end to the bone, this would all be the muscle tendon. So the muscle tendon is made up of endomysium, paramysium, and epimysium. But now we do not have the muscle cells. That's why the muscle tendon is more narrow than the muscle belly. The tendon then inserts onto the bone. And the next question would be, how does the tendon actually insert onto the bone? Is there some type of anatomical glue that's holding it together? Well, if you think back to our discussion on osteology, and you remember what was the tissue that made up the extracellular matrix of bone? Well, it was mineralized collagen, or collagen and hydroxyapatite. What this means is that the bone has a collagen component itself. So the tendon of the muscle blends into the collagen component of bone. So we can see the paramycium, and really the endomycium should continue on as well. So the endomycium and the paramycium dive directly into the bone, and the collagen of the tendon becomes one with the collagen of the bone. And then the epimycium comes, and you can see it sort of laying out over the surface of the bone. And if we think about what is on the surface of the bone, well, it's periosteum. And periosteum is made up of dense, irregular, collagenous connective tissue. So the epimycium portion of the tendon blends into the periosteum of the bone. So in this way, the connective tissue that surrounds the muscle fibers extends beyond the muscle fibers to become the tendon, and then that same collagen becomes one with the collagen of our bony structures, and that's how the tendons actually attach to the bone. Now we're going to begin looking a little bit closer at the anatomy of an individual muscle cell. And the first thing that I want everyone to realize is that muscle cells are similar to other cells throughout the human body. In other words, they have things like mitochondria, they have nucleus. In this case, muscle cells are multinucleated. They have a cell membrane, which we call the sarcolemma. They have an endoplasmic reticulum that we call a sarcoplasmic reticulum, and so on and so forth. So very similar to other cells found throughout the human body. But what makes a muscle cell unique is that it has a specialized arrangement of contractile proteins within the muscle cell that allows it to develop tension. These contractile proteins are arranged into groupings that are known as sarcomeres, and it's the appearance of the sarcomere that gives the muscle cell its striated appearance. Here we see an image of 3 individual skeletal muscle cells, and you can see those purple blobs that are the nuclei of the different muscle cells. And then that alternating light and dark striped pattern across the muscle cells are what are termed the striations. And again, these striations are produced by the arrangement of sarcomeres, the contractile element within the skeletal muscle. So now, jumping from this image, let's take a look at a more diagrammatic image and break it down a little bit. All right, so here we see an individual muscle cell. So you can take it from sort of its macro point. Here we have a full muscle, and within that muscle we see individual fascicles. So this here would be a single fascicle. And remember, a fascicle are individual muscle cells and their endomycium, and then packaged by a wrapping of paramycium. So that would create this fascicle. Then if we zoom in on just one single muscle cell, that's what has been blown up in this image. So starting from the outside, this sort of clear looking wrapping, that would be the sarcolemma, or the cell membrane. When we cut away the sarcolemma and look inside, we find multiple little pillars that are contractile proteins, and these are called myofibrils, all of these individual pillars of contractile proteins. And when we zoom in on an individual myofibril, now we see that there are myofilaments that make up a myofibril. And the myofilaments, these are the thick and thin filaments where we find the proteins actin and myosin. Also notice how even in this diagrammatic image, we have a portion that is darker in the myofibril and a portion that is lighter. And the dark portions and light portions of one myofibril line up with the dark portions and light portions of the next myofibril, hence giving it that dark to light striated appearance that we find in skeletal muscle. You also notice the nuclei, again, multinucleated cells here. We have mitochondria represented throughout the muscle, and the sarcoplasmic reticulum shown here in blue. And later on in class, when we talk about how we excite a skeletal muscle cell, we'll talk a lot more about this structure shown here in green, which are the transverse tubules of the skeletal muscle cell. So now the next thing that we're going to do is we are going to talk about the individual element of the muscle cell, the individual contractile element known as a sarcomere. This image is as if we have gone inside of a skeletal muscle cell and we're looking at two individual myofibrils from a longitudinal view. And what we can see is again that classic dark band, light band, dark band, light band striation of the skeletal muscle. And what has been shown down here on the bottom is from one of these dark lines to the next dark line, that unit that runs between is what is known as an individual sarcomere. So let's break down this sarcomere a little bit more and learn the anatomy of an individual sarcomere. Here we see a diagram of an individual sarcomere. The sarcomere is made up of thick and thin filaments. The thick filaments in the image below are the portion shown in green right in the center of the sarcomere. The thin filaments are the portion shown in purple. Now, there are a number of different proteins that are found in an individual sarcomere, but for this class we are only going to discuss the primary proteins. So let's take a look at the primary proteins on the thick filaments. There are two primary proteins that make up the thick filament. The green portion shown below is the myosin. This is where you see those individual little lobules, those little heads. Those are the myosin heads that are actually going to be the portion of the sarcomere that move. Those are the little motors of the sarcomere. The titin molecule is the yellow band that runs right down through the center of the thick filament. And then when the thick filament ends, the titin molecule continues on all the way to this sort of zigzagging portion that marks the end of the sarcomere. For the thin filaments, we have three primary proteins. The small purple spheres that you see in the image below that are linked together in a helical pattern are the actin molecules. The actin molecules contain the binding site for the myosin head to grab onto in order to actually pull on the thin filament and shorten a sarcomere. Also on the thin filament are two regulatory proteins, troponin and tropomyosin. Tropomyosin blocks the myosin binding sites on actin, so it prevents us from being able to contract a muscle. Troponin regulates tropomyosin. It can allow tropomyosin to continue to block the binding sites, or when stimulated, troponin will move tropomyosin away from those binding sites to allow the myosin heads to interact with actin, and this is what initiates the increase in tension within a skeletal muscle cell. The next thing that we need to do is identify the anatomy of an individual sarcomere. And I've arranged it so that we just look at two elements at a time. And the first two elements that I want to point out are the I bands and the A bands. The I bands are the region of the sarcomere that contain thin filaments, but no thick filaments whatsoever. The A bands are essentially everything else. So the A bands are where we find all of the thick filaments, and there is some thin filament overlap. So let's look at this on an image. So first, on this image, on the bottom, we see a diagrammatic image of sarcomeres, and above we see a microscopic image of sarcomeres. And I have the elements of interest pointed out on each of these two images. So if we look at the diagrammatic image, remember the I band was where we have thin filaments, but no thick filaments. So that accounts for the lighter portion of a sarcomere, seen here. The A bands are where we find all of the thick filaments. So the A band shown here is from one end of the thick filament all the way to the other end. And what you'll notice is that the A band does contain some thin filament overlap. If you look at the image up above, you see the I band is the very light portion of the sarcomere, and here we're actually crossing from one sarcomere to the next sarcomere. So the I band is the entire light portion, so it has parts of both sarcomeres in it. The A band is the darker band because that's where all of the thick filaments are. Next, we have the H bands and the M lines. The H bands are the center of the A band. So just a quick recap, the A band is where we find the thick filaments. So the H band is the very center of the A band, and it's the portion of the A band where there are no thin filaments. The M line runs right down the center of each A band, and it helps anchor the thick filaments. So again, let's take a look at this on the diagrams. So on the diagram at the bottom, remember the A band covered the entire thick filament. The H band is the center of the thick filament where there is no thin filament overlap. So here's the H band. The M line runs right down through the center of the A band and H band here, anchoring the thick filaments. So if you look up at the image above, the H band is this lighter portion right in the middle of the A band. And it's lighter because there are no thin filaments there, so it's less dense. The M line is this dark line that goes right down through the middle of it. You can see it right over here. Okay, the dark line right down through the middle of the A band. The last piece of sarcomere anatomy that we need to identify are the Z discs. The Z discs are found in the center of each I band. The I band being the lighter portion where we have thin filaments and no thick filaments. And the region from one Z disc to the next Z disc is what defines an individual sarcomere. So here we see the Z disc as this zigzagging blue chain here that the thin filaments are anchoring to. And we have a Z disc on one side and a Z disc on the other side, and the region from one Z disc to the next is what we call the sarcomere. If you look up at the image above, we see that dark band of the Z disc right in the middle of the I band on one side and on the other side. So this would define a single sarcomere. On this slide, I've put all of the anatomical elements of the sarcomere back in on the image so that you can use this image to study from. I do want all of you to be able to draw a sarcomere and label all of the anatomy of an individual sarcomere. Now, so far, we've looked at the sarcomere from more of a two-dimensional sort of longitudinal section view. But what I want to remind you all of is that sarcomeres are contained within myofibrils, and myofibrils are like little columns of contractile proteins. So they are three-dimensional in shape. So on the next slide, we're going to take a quick look at the three-dimensional shape of sarcomeres. So here we again see an individual skeletal muscle cell with multiple myofibrils contained within. And if we pull out an individual myofibril and zoom in on it, that's what we see here. So if you look just straight in on it, we see the sort of standard two-dimensional structure that we're used to of looking at a sarcomere. But realize that this is a column containing multiple sarcomeres arranged three-dimensionally in relation to one another. The regions below are cross-sections through this myofibril at different points. So if we look at, for example, the M line, which is the center of the H band and A band, all that we see here are the cross sections of the thick filaments and the proteins that are anchoring the thick filaments to one another. That's what creates the M line. If we move out from the M line, so we get into the H band region here where we have thick filaments but no thin filaments, this is what we would see in cross section. So again, we're seeing the cross section of the thick filament, but we don't see any thin filaments. And by the way, that little green dot that's in the middle, that would be the titin molecule that is running down through the center of the thick filament. If we move on to the next region, so now we're in the portion of the A band where we have some thin filament overlap, and we look at that in cross section. Well, now we can see a thick filament in cross section with the titin molecule in the middle, and surrounding each thick filament, we see 6 thin filaments. 1, 2, 3, 4, 5, 6 thin filaments. This is because a thick filament's myosin heads are arranged 360 degrees around the thick filament so that one thick filament can interact with 6 thin filaments in order to generate tension within the muscle. If we go outside of the A band, so now we're in the I band, and we look at this region of the I band where we have thin filaments and no thick filaments, it would look like this in cross-section. So notice how we no longer see the purple cross-section of the thick filament, but we do still see the green cross-section of the titin molecule. This is because, remember, the titin molecule extends past the thick filament and attaches to the Z-disc, so we can still see it here. We also see the cross-section of the individual thin filaments. And finally, if we get all the way out to the edge of a sarcomere and we take a cross-section right at the Z-disc, we see the titin molecules attaching to the Z-disc, the thin filaments attaching to the Z-disc, and all of these supporting protein structures that run between them that help create the Z-disc formation. So again, the take-home from this image is just to remember that sarcomeres are not two-dimensional. They are a three-dimensional structure within a myofibril. And on the next image, I'll actually show you a cross-section of individual myofibrils to bring this idea home. First, let me orient you on this image. So we're looking at a cross-section, and in this cross-section, we see portions of 1, 2, and even a little bit of a third myofibril peeking in here. This structure, these sort of darker areas that are around the myofibrils, those are mitochondria. And if we look at this one's a little bit more clear. The more thick, darker circles are an individual thick filament shown in cross-section. And you notice surrounding that thick filament, we have 1, 2, 3, 4, 5, 6 thin filaments. This is what forms that hexagonal arrangement, where there's 6 thin filaments surrounding an individual thick filament. So this cross-section would be somewhere in the A band where we have thin filament overlap with the thick filaments. And the last topic that I want to discuss in this audio lecture is how sarcomeres change as we undergo a shortening contraction of a skeletal muscle. So this is what's called the sliding filament theory. The basic idea of the sliding filament theory is that the thin filaments are going to slide along the thick filaments in order to shorten a sarcomere. So the first concept that I want you to have down to understand the sliding filament theory is that the thick filaments are our anchor point, or our point of reference. So you can imagine the M line, that very center of the A band, as the point within the sarcomere that is not moving. And what we're going to do is the myosin heads of the thick filament are going to grab onto the actin of the thin filament, and they are going to pull on the thin filaments and slide the thin filament along the thick filament. What this will do is it will pull the Z-discs closer to the M line. And as the Z-discs are pulled closer to the M line, this is going to make both the H band and the I band become smaller. So if you look at the example images down below, we see sarcomeres lined up end to end here. Okay, so we have a set of sarcomeres here, a set of sarcomeres here, and they're arranged longitudinally, lined up end to end. This is our resting condition where we see our I band, the lighter portion, where we have thin filaments but no thick filaments, our A band containing the thick filaments, our H band containing thick filaments but no thin filament overlap. And as the muscle develops tension and the thin filaments slide along the thick filaments and the sarcomere shortens, you notice that the regions known as the I band and the H band are getting smaller and smaller. So the takeaway message here, sliding filament theory, the thick filaments are the anchor point, they are the motors, and they pull on the thin filaments like little ropes to draw the Z-discs towards the M line in order to shorten the sarcomere and therefore shorten the overall length of a skeletal muscle cell. And that concludes the audio lecture looking at the structure of human skeletal muscle.